Today we're going to talk about the rate of photosynthesis, and the goal of this video is to describe how you can measure the rate of photosynthesis and identify factors that can affect the rate of photosynthesis. So, quick recap here. There are two steps of photosynthesis. The first is called the light-dependent step. We call this the light-dependent step because light is needed in order for this step to occur. This step happens in the thylakoid, which are those green discs within the chloroplast. The second step of photosynthesis is called the light-independent step. This is also known as the Calvin cycle, so you can call it both names. We call this the light-independent step because light is not needed for this step to occur. This step can occur in the dark. And this step happens in the stroma, which is that space outside of the thylakoid that's filled with fluid. So those are the two steps of photosynthesis. Now, there are inputs and outputs of each step. To understand what those inputs and outputs are, it's helpful to look at the chemical equation for photosynthesis. So, carbon dioxide and water are the reactants in photosynthesis, and glucose and oxygen are the products. But like I said, there are specific inputs and specific outputs of each step of photosynthesis. So let's look at those in a chart. For the light-dependent step, that's the step that occurs in the thylakoid, water and sunlight go in, and oxygen comes out. So water and sunlight go into the thylakoid, and oxygen comes out of the thylakoid. That is the light-dependent step. The light-independent step has carbon dioxide go in and glucose come out. So those are the inputs and outputs of each step. So that's just a brief recap of what's going on during photosynthesis. If we were to put all of this into one image, it would look like this. You have water and sunlight going into the thylakoid and oxygen coming out. That's the light-dependent reaction. And you have carbon dioxide going into the stroma and glucose coming out. That is the light-independent reaction, which is also known as the Calvin cycle. And notice the thing that connects these two steps is something called ATP and NADPH. So ATP is an energy-carrying molecule, and so is NADPH. And so those molecules carry energy to the stroma where the Calvin cycle can occur, and that energy is going to be used to make glucose there. And then ADP and NADP plus go back to the thylakoid where they can use more energy from sunlight and continue this process. Now, how do you measure the rate of photosynthesis? You can do that by having a setup like the one here in the picture. Let me briefly describe it. Take a plant and place it underwater. Have it at some specific distance from a light source. So here you can see there's a ruler. Then you turn the lamp on so it has light, and you can begin counting the number of bubbles that get produced by that plant in some set amount of time. So you'll need to have a stopwatch, and you keep track of the time. Maybe every minute you're counting how many bubbles are produced in that minute. Now, why do we care about the bubbles? Well, the bubbles indicate that the plant is producing oxygen. So when that plant is underwater and it gives off the gas, namely oxygen, a bubble is produced. And so that indicates that photosynthesis is happening. If oxygen is being produced by the plant, we know that photosynthesis is occurring. If lots of bubbles are produced, then you have a very high rate of photosynthesis. If very few bubbles are produced, then you have a low rate of photosynthesis. We'll finish up by talking about factors that can affect the rate of photosynthesis here. So I'm going to list 4, and you'll probably notice something about these 4 factors. One of them is the intensity of the light. So if you move the lamp closer to the plant, that can intensify the light. Or if you increase the brightness of the light, that can increase the light intensity as well. And notice notice something in the graph here. If the light intensity increases, then the rate of photosynthesis increases. However, you reach a kind of maximum point, a threshold. And above that, even if you continue to make the intensity of the light greater, so make the light brighter and brighter and brighter, at some point it just won't increase the rate of photosynthesis anymore. So that's one factor that can affect the rate of photosynthesis. A second factor is carbon dioxide concentration. So that's this middle graph. As you increase the amount of carbon dioxide that's available to the plant, the rate of photosynthesis increases. But just like light intensity, you reach a point where even if you continue giving the plant more and more and more carbon dioxide, it will not increase the rate of photosynthesis beyond that point. So both of these, light intensity and carbon dioxide, are inputs of photosynthesis. Temperature is a third factor that can affect photosynthesis. So notice in this graph down here, as the temperature increases, the rate of photosynthesis increases. However, there is a kind of peak temperature, and beyond that point, if it gets any hotter, then the rate of photosynthesis begins to decrease. So for plants, there's a kind of optimal temperature that they do photosynthesis. The same goes for water. I don't have a graph to represent this, but if you've ever watered a plant, you may have noticed if you give it too little water, it ends up wilting and dying. But if you also give it too much water, the same thing can happen. So temperature and water have something in common here. There's a kind of optimal level, and too little or too much can actually decrease the rate of photosynthesis. So if I were to sum this up, these factors, light intensity and carbon dioxide concentration, here's what they have in common. Both can increase the rate of photosynthesis, but both will reach a threshold that they can't go past. The rate of photosynthesis can't increase beyond that point. Temperature and water, on the other hand, can increase the rate of photosynthesis, but they reach an optimal amount. Beyond that point, they can decrease the rate of photosynthesis. So to sum up, you've seen the inputs and outputs of photosynthesis, and you've seen how to measure the rate of photosynthesis, and you've seen some factors that can affect the rate of photosynthesis.